Hi, this is Dr. Cassandra Quave, and in today's lecture we'll be covering Chapter 4 from your textbook, discussing the characteristics of prokaryotic and eukaryotic cells. So all living cells can be classified as either prokaryotic or eukaryotic. Prokaryotic cells lack a nucleus and other membrane-enclosed structures, whereas eukaryotic cells have such structures. Here's a useful table from your textbook that goes over many of the different differences between prokaryotic and eukaryotic cells. All prokaryotes are single-celled organisms, and all are bacteria. Most of your book will be devoted to the study of prokaryotes. Eukaryotes, on the other hand, include all plants, animals, fungi, and protists. Prokaryotic and eukaryotic cells are similar in several ways. For example, both are surrounded by a cell membrane or a plasma membrane. Although some cells have structures that extend beyond this membrane or surround it, the membrane defines the boundaries of the living cell. Both prokaryotic and eukaryotic cells also encode genetic information in DNA molecules. These two types of cells, however, are different in other important ways. For example, in eukaryotic cells, DNA is found in a nucleus surrounded by a membranous nuclear envelope. But in prokaryotic cells, DNA is a nuclear region not surrounded by a membrane. Eukaryotic cells also have a variety of internal structures known as organelles, or little organs, that are surrounded by one or more membranes. Prokaryote cells generally lack organelles that are membrane enclosed. So what you should do on this table is to closely go through and compare some of the similarities and differences between prokaryotic and eukaryotic cells. And here's the continuation of this same table, looking at extracellular structures regarding things like the cell wall. For example, in prokaryotic cells, you have peptidoglycan found in most, whereas eukaryotes have cellulose, chitin, or both, found in plant and fungal cells. And then discussion of other structures like flagella, cilia, and pili. Likewise, the reproductive processes of both cells differs. In prokaryotic cells, they undergo binary fission, whereas in eukaryotes undergo mitosis and/or meiosis. So again, use this as a useful study tool as you go through this chapter. Let's go in detail a bit more about prokaryotic cells, discussing their size, their shape, and their arrangement. All members of the archaea and bacteria are prokaryotes and have been traditionally called types of bacteria. Prokaryotes are among the smallest of all organisms. In fact, most prokaryotes range from 0.5 to 2 micrometers in diameter. As a point of comparison, a human red blood cell is about 7.5 micrometers in diameter. Because of their small size, bacteria also have a large surface-to-volume ratio. For example, spherical bacteria with a diameter of 2 microns have a surface area of about 12 micrometers squared and a volume of about 4 micrometers cubed. And their surface-to-volume ratio is 12 to 4, or 3 to 1. In contrast, eukaryotic cells that have a diameter of 20 millimeters have a surface area of about 1200 micrometers squared and a volume of 4000 micrometers cubed. And their surface-to-volume ratio is 1,200 to 4,000, or 0.3 to 1, only 1/10 as great as that of the prokaryotes. This large surface-to-volume ratio of bacteria means that no internal part of the cell is very far from the surface, and that nutrients can easily and quickly reach all parts of the cell. Now, regarding shape, typically bacteria come in 3 basic shapes: spherical, rod-like, and spiral. But there are many variations that abound. A spherical bacterium is called a coccus. A rod-like bacterium is called a bacillus. Some bacteria, called coccobacilli, are short rods, intermediate in shape between a cocci and bacilli. Spiral bacteria have a variety of curved shapes. A comma-shaped bacterium is called a vibrio, and a rigid, wavy-shaped one is a spirillum, and a corkscrew-shaped one is a spirochete. Now, with regards to arrangement, in addition to their many characteristic shapes, many bacteria also are found in distinctive types of groupings. These groups form when the cells divide without separating. Cocci can divide into one or more planes or randomly. Division on one plane produces cells in pairs, and these are known with the prefix diplo, or in chains with the prefix strepto. Division in two planes, however, produces cells in tetrads, or 4 cells arranged in a cube, as shown here. Division in 3 planes produces sarcini. Random division planes produce grape-like clusters called staphylo. Bacilli can divide only in one plane, but they can produce cells connected end-to-end, kind of like train cars, or side by side. Spiral bacteria are not generally grouped together, but found individually. An important thing to note is that prokaryotes divide by binary fission rather than by mitosis or meiosis. New cell wall material grows and the cell pinches in half through this area. Inside, the chromosome is duplicated and one is found in each daughter cell. Let's go through the structure of a typical prokaryotic cell. Bacterial cells consist of the following parts, typically. You have a cell membrane that is usually surrounded by a cell wall and sometimes by an additional outer layer. There's an internal cytoplasm with ribosomes, a nuclear region, and in some cases granules and/or vesicles. And there's a variety of external structures such as capsules, flagella, and pili. Now let's look in detail at the cell wall. The semi-rigid cell wall lies outside the cell membrane in nearly all bacteria, and it performs 2 important functions. It maintains the characteristic shape of the cell, and it prevents the cell from bursting when fluids flow into the cell by osmosis. Now let's talk in more detail about peptidoglycan in the cell wall, these different components. Peptidoglycan is the single most important component of the bacterial cell wall. It is a polymer so large that it can be thought of as one immense covalently linked molecule, and it forms a supporting net around a bacterium that resembles multiple layers of a chain-linked fence. Gram-positive cells may have as many as 40 layers of peptidoglycan. Cell walls of gram-positive organisms have an additional molecule called teichoic acid, which consists of glycerol, phosphates, and sugar alcohol ribitol, and occurs in polymers up to 30 units long. Now, regarding the outer membrane, this is a part found primarily in gram-negative bacteria, and it's a bilayer membrane. It forms the outermost layer of the cell wall and is attached to the peptidoglycan by an almost continuous layer of small lipoprotein molecules. Lipopolysaccharide, also known as endotoxin or LPS, is also an important part of the outer membrane, and this can be used to identify gram-negative bacteria. It's an integral part of the cell wall and is not released until the cell walls of dead bacteria are broken down. Lipopolysaccharide consists of polysaccharides and lipid A. The polysaccharides are found in repeating side chains that extend outward from the organism. The lipid A portion is responsible for the toxic properties that make many gram-negative infections a potentially serious medical problem. Lipopolysaccharide can cause fever and dilated blood vessels, so it can result in a dramatic drop in blood pressure. Now, the periplasmic space is another distinguishing characteristic of many bacteria, and this is a gap found between the cell membrane and the cell wall. The gap is most easily observed through electron microscopy of gram-negative bacteria. In these organisms, the gap is again called the periplasmic space. and it represents a very active area of cell metabolism. This space contains not only the cell wall peptidoglycan, but also many other digestive enzymes and transport proteins that can destroy potentially harmful substances. Periplasmic spaces are rarely observed in gram-positive bacteria. However, such bacteria must accomplish many of the same metabolic and transport functions that gram-negative bacteria do. At present, most gram-positive bacteria are thought to have only periplasms, but not the periplasmic space, where their metabolic digestion occurs and new cell wall peptidoglycan is attached. Now let's look specifically at the gram-positive cell wall. The cell wall in gram-positive bacteria has a relatively thick layer of peptidoglycan, as we previously mentioned, that can be from 20 to 80 nanometers across. This peptidoglycan layer is closely attached to the outer surface of the cell membrane. If peptidoglycan is digested from the cell wall, gram-positive bacteria become something known as a protoplast, or cells with a cell membrane but no cell wall. Protoplasts shrivel or burst unless they're kept in an isotonic solution, which is a solution that has the same pressure as that inside the cell. The thick walls of gram-positive bacteria retain stains like crystal violet in the cytoplasm, but yeast cells, many of which have thick walls but no peptidoglycan, also retain these stains. So, retention of gram stains seems to be directly related to wall thickness and not exactly to peptidoglycan. Gram-positive bacteria lack both an outer membrane and a periplasmic space, so digestive enzymes are not retained in the periplasm, are released into the environment, where they sometimes become so diluted that the organisms derive no benefit from them. Now let's discuss the gram-negative cell wall. The cell wall of a gram-negative bacterium is thinner but much more complex than that of a gram-positive bacterium. Only 10 to 20% of the wall is peptidoglycan. The remaining portion consists of various polysaccharides, proteins, and lipids. The cell wall also contains an outer membrane, which constitutes the outer surface of the wall, leaving only a very narrow periplasmic space. The inner surface of the wall is separated from the cell membrane by a wider periplasmic space, and this is where toxins and enzymes remain in sufficient concentrations to help destroy substances that might harm the bacterium, but they do not harm the organism that produces them. If the cell wall is digested away from a gram-negative bacterium, it becomes a spheroplast, which has both a cell membrane and most of the outer membrane. Gram-negative bacteria fail to retain the crystal violet iodine dye during the decolorizing procedure, partly because of their thin cell walls and partly because of the relatively large quantity of lipoproteins and lipopolysaccharides in the wall. Regarding other kinds of cell walls, let's discuss the acid-fast bacteria. Although the cell wall of acid-fast bacteria, also known as the mycobacteria, is very thick, like that of the gram-positive bacteria, it is, however, approximately 60% lipid and contains much less peptidoglycan. In the acid-fast staining process, carbonyl fusion will bind to the cytoplasm and resist removal by an acid-alcohol mixture. The lipids make acid-fast organisms impenetrable to most other stains and protects them from acids and alkalis. These organisms grow very slowly because the lipids impede entry of nutrients into the cells, and the cells must expend large quantities of energy to synthesize lipids. Now, regarding wall-deficient organisms, bacteria that belong to the genus Mycoplasma have no cell walls. They're protected from osmotic swelling and bursting by a strengthened cell membrane that contains sterols. These are molecules typical of eukaryotes and are rarely found in prokaryotes. However, the protection is not complete, and often mycoplasmas must be grown in special media. Without a rigid cell wall, they vary widely in shape, often forming slender branched filaments and exhibiting extreme pleomorphisms. Other genera of bacteria that may normally have a cell wall but can suddenly lose their ability to form their cell wall These are wall-deficient strains called L forms, named after the Lister Institute where they were discovered over 70 years ago. This loss of cell wall may occur naturally or be caused by chemical treatment, and L forms play a role in chronic or recurrent diseases. Treatment with antibiotics that affect cell wall synthesis will kill most of the bacteria in some infections, but it can leave a few alive as the L forms. When treatment is discontinued, those L forms can then revert to walled forms and regrow an infecting population. An example of this is found in the association of the bacterium Mycobacterium paratuberculosis with Crohn's disease, which is a chronic disorder of the intestine. Some archaea may actually entirely lack cell walls, while others have unusual walls of polysaccharides or of other proteins, but lack true peptidoglycan. Instead, they may have a similar compound called pseudomurine. Now let's talk about the prokaryotic plasma membrane. A cell membrane, or the plasma membrane, is a living membrane that forms the boundary between a cell and its environment. Also known as the cytoplasmic membrane, this dynamic, constantly changing membrane is not to be confused with the cell wall. Bacterial cell membranes have the same general structure as membranes of other cells, and such membranes consist mainly of phospholipids and proteins. The fluid mosaic model represents the current understanding of the structure of such a membrane. The model's name is derived from the fact that phospholipids in the membrane are in a fluid state and that proteins are dispersed among the lipid molecules in the membrane, forming a mosaic pattern. And here you can see a picture of the basic structural component of the membrane, which is the phospholipid molecule. The phospholipid molecule has two long fatty acid tails of hydrocarbon, and these tails are very hydrophobic, or they do not interact with water, and they form an oily barrier to most water-soluble substances. The head of the molecule consists of a charged phosphate group, usually joined to a charged nitrogen-containing group, and the head is very hydrophilic, interacting with water. Here you can see a picture of the prokaryotic plasma membrane based off of the fluid mosaic model. The phospholipids form a bilayer in which the hydrophobic tails may form the central core, and the hydrophilic heads, or the water-loving heads, form the surfaces that face both the interior of the cell and the outside environment. In this fluid bilayer, proteins float like icebergs. Some extend through the bilayer, whereas others are anchored to the inner or outer surface. Proteins and membrane lipids to which carbohydrate chains are attached are called glycoproteins and glycolipids. A few bacteria, like the mycoplasmas, have cholesterol molecules in their cell membranes, as do most eukaryotes. However, mycoplasmas lack cell walls, and cholesterol molecules add rigidity to the cell membrane. With regards to internal structures, bacterial cells typically contain ribosomes, a nucleoid, and a variety of vacuoles within their cytoplasm. Some bacteria also sometimes contain endospores as well. The cytoplasm of prokaryotic cells is the semi-fluid substance found inside the cell membrane. Because these cells typically only have a few clearly defined structures, such as a few chromosomes, some ribosomes, they mainly consist of this material, the cytoplasm. Cytoplasm is about 4/5 water and 1/5 substances dissolved or suspended in the water, and these substances include enzymes and other proteins, carbohydrates, lipids, and a variety of inorganic ions. Ribosomes consist of RNA and protein, and they are abundant in the cytoplasm of bacteria, often grouped in long chains called polyribosomes. Ribosomes are nearly spherical, stain densely, and they contain a large subunit and small subunit. Ribosomes serve as sites for protein synthesis in the cell. Whole bacterial ribosomes, which are smaller than eukaryotic ribosomes, have a rate of 70S. Their subunits have rates of 30S and 50S. And the S refers, of course, to the sedimentation rate, or the rate at which it moves to the bottom of a tube. One of the key features differentiating prokaryotic cells from eukaryotic cells is the absence of a nucleus bounded by a nuclear membrane. Instead of a nucleus, bacteria have a nuclear region known as a nucleoid. The centrally located nuclear region consists mainly of DNA, but has some RNA and protein associated with it. Here you can see a picture of a bacterial nuclear region, and this is an image taken through transmission electron microscopy, and it's a thin section of Escherichia coli with the DNA stained in red. Now let's talk about inclusions. Bacteria can have within their cytoplasm a variety of small bodies that are collectively known as inclusions. Some are called granules and others are called vesicles. Granules, although they're not bounded by a membrane, contain substances that are so densely compacted that they do not easily dissolve into the cytoplasm. And each granule contains a specific substance, such as glycogen or polyphosphate. Glycogen is a glucose polymer that is used for energy, whereas polyphosphate is a phosphate polymer that supplies the phosphate for a variety of metabolic processes. Polyphosphate granules are called volutin or metachromatic granules because they display metachromasia. That is, although most substances stained with a simple stain such as methylene blue take on uniform solid color, metachromatic granules exhibit different intensities of color. Certain bacteria also have specialized membrane-enclosed structures called vesicles. Some aquatic photosynthetic bacteria and cyanobacteria have rigid gas-filled vacuoles. These organisms regulate the amount of gas in the vacuoles and therefore the depth at which they float to obtain optimum light for photosynthesis. Now let's discuss endospores. The properties of bacterial cells that we've just described pertain to something known as vegetative cells, or cells that are metabolizing nutrients. However, vegetative cells of some bacteria, such as Bacillus or Clostridium, can produce resting stages called endospores. Although bacterial endospores are commonly referred to as spores, do not confuse them with fungal spores. A bacterium produces a single endospore, which merely helps that organism survive and is not a means of reproduction, whereas a fungus produces numerous spores that help the organism survive and provide a means of reproduction. Bacterial endospores are formed within the cell, and you can see here a picture of the green stained endospore inside the cell. What's unique about endospores is that they contain very little water and are highly resistant to heat, drying, acids, bases, certain disinfectants, and even radiation. So they're very difficult to harm. The depletion of a nutrient is usually enough to induce a large number of cells to produce spores.
We are unavailable
However,
to take your call.
many
Please leave a message. Thank you for calling.
However, many investigators believe that spores are part of the natural life cycle and that a few are formed even when nutrients are adequate and environmental conditions are favorable. Thus, sporulation or endospore formation seems to be a means by which some bacteria prepare for the possibility of future adverse conditions. In other words, they're a good survival mechanism. Endospores, in fact, are capable of surviving adverse environmental conditions for long periods of time, some for over 10,000 years. Spores of Antarctic bacteria can remain dormant for at least 10,000 years at a temperature of minus 14 degrees in ice 430 meters deep, whereas others can withstand hours of boiling. When conditions become more favorable, the endospores will go through a process known as germination, or begin to develop into functional vegetative cells. Because endospores are so resistant, there are special methods that are necessary in order to kill them during sterilization. Otherwise, they will germinate and grow in the media that's thought otherwise to be sterile. Now let's let's discuss some of the external prokaryotic structures. In addition to cell walls, many bacteria have structures that extend beyond or surround the cell wall. Flagella and pili extend from the cell membrane through the cell wall and beyond it. Capsules and slime layers surround the cell wall. About half of all known bacteria are motile, or capable of movement. They often move with speed and apparent purpose, and they usually move by means of long, thin helical appendages known as flagella. A bacterium can have one flagellum or two or many flagella. Bacteria with a single polar flagellum located at one end or pole are called monotrichous. Bacteria with two flagella, one at each end, are amphitrichous. Bacteria with two or more flagella at one or both ends are called lophotrichous, and those with flagella all over the surface are peritrichous, such as in the picture shown here on the right. Most flagella work by rotating like twirling L-shaped hooks, such as a dough hook on a kitchen mixer, or the rotating string of a hand-carried grass trimmer. Motion is thought to occur as energy is used to make one of the rings in the cell membrane rotate with respect to the other. When flagella bundle together, they can rotate counterclockwise and the bacteria run or move in a straight line. When the flagella rotate clockwise, however, the flagellar bundle comes apart, causing the bacterium to tumble randomly. Both runs and tumbles are generally random movements. Let's talk a bit about chemotaxis and motility behaviors now. Sometimes bacteria move toward or away from substances in their environment by a non-random process known as chemotaxis. Concentrations of most substances in the environment vary along a gradient, that is, from high to low concentration. When a bacterium is moving in the direction of increasing concentration of an attractant, such as a nutrient they want to get to, it tends to lengthen its runs and to reduce the frequency of its tumbles. However, when it is moving away from the attractant, it shortens its runs and increases the frequency of its tumbles. So even though the direction of the individual runs is still random, the net result is movement towards the attractant, or positive chemotaxis. Movement away from the repellent, or negative chemotaxis, results from the opposite responses. Now let's discuss some of the other parts of axial filaments. Or actually, I'm going to go to pili, excuse me. Let's discuss pili. So pili are tiny hollow projections that are used to attach bacteria to surfaces and are not involved in movement. So this is different from flagella. A pilus is composed of subunits of the protein pilin. Bacteria can have two types of pili, long conjugation pili, or F. pili, also called sex pili, and short attachment pili, also known as fimbriae. Conjugation pili, or sex pili, are found in only certain groups of bacteria, and they can attach two cells and may furnish a pathway for the transfer of genetic information between the cells. And this process of transfer of information is known as conjugation. Transfer of DNA furnishes genetic variety for bacteria, as sexual reproduction does for many other life forms. And such transfers among bacteria cause problems for humans because of antibiotic resistance that can be passed on with DNA transfer. Now, attachment pili, or fimbriae, serve a different function, and they help bacteria to adhere to surfaces, such as cell surfaces and the interface of water and air. They contribute to pathogenicity of certain bacteria, or their ability to produce disease, by enhancing colonization. For example, some bacteria adhere to red blood cells by attachment pili and cause the red blood cells to clump, in a process known as hemagglutination. In some species of bacteria, some individuals have the attachment pili and others lack them. For example, in Neisseria gonorrhea, the causative agent of gonorrhea, strains without pili are rarely able to cause gonorrhea, but those with pili are highly infectious because they attach to epithelial cells of the urogenital system, and these pili allow them to attach to sperm cells and thereby spread on to the next individual. Now let's discuss the glycocalyx. Glycocalyx is a term used to describe all polysaccharide-containing substances found external to the cell wall, from the thickest capsules to the thinnest slime layers. All bacteria have at least a thin slime layer. A capsule is a protective structure found outside of the cell wall of the organism that secretes it. Only certain bacteria are capable of forming capsules, and not all members of a species have capsules. For example, the bacterium that causes anthrax does not produce a capsule when it grows outside of an organism, but it does when it infects an animal. Capsules are typically made up of complex polysaccharide molecules that are arranged into a loose gel. However, the chemical composition of each capsule is unique to the strain of the bacteria that secretes it. Now, a slime layer, on the other hand, is less tightly bound to the cell wall and is usually thinner than a capsule. When present, the slime layer protects the cell against drying. It also helps it to trap nutrients near the cell and can sometimes bind the cells together. Slime layers allow bacteria to adhere to objects in their environment, such as rock surfaces or the root hairs of plants, so that they can remain near sources of nutrients or oxygen. This biofilm protects bacteria on the bottom of the layers from environmental or man-made chemicals. For example, some oral bacteria can adhere by their slime layers and form dental plaque on the teeth. The slime layer keeps the bacteria in close proximity to the tooth surface, where it can then cause dental caries. Now let's move on to eukaryotic cell structure so that we can differentiate this from the prokaryotes that we've just discussed. Eukaryotes are larger and more complex than prokaryotic cells. Most eukaryotic cells have a diameter of more than 10 micrometers, and many are much larger. They contain a variety of highly differentiated structures. These cells are the basic structural unit of all organisms in the kingdoms Protista, Plantae, Fungi, and Animalia. Eukaryotic organisms also include microscopic protozoa, algae, and fungi, and thus are appropriately considered in microbiology. Here you can see a picture of the general structure of a eukaryotic cell. Let's discuss some of the core components to the eukaryotic cell, starting with the plasma membrane. The cell membrane or plasma membrane of a eukaryotic cell has the same fluid mosaic structure as that of a prokaryotic cell. In addition, eukaryotes also contain several organelles enclosed by membranes that have similar membrane structure. Eukaryotic membranes differ from prokaryotic membranes in some respects, especially in the great varieties of lipids that they contain. They also contain sterols, found among prokaryotes only in the mycoplasmas. Sterols help to add rigidity to the membrane, and this can be important in keeping the membranes intact in eukaryotic cells. Because of their large size, eukaryotic cells have a much lower surface-to-volume ratio than prokaryotic cells. Now, the internal structure of a eukaryotic cell is much more complex than a prokaryote. With regards to the cytoplasm, this makes up a relatively smaller portion of the eukaryotic cells than that of the prokaryotes because of the nucleus and many organelles that fill up the space in the cell. Like the cytoplasm of prokaryotic cells, the cytoplasm of the eukaryotic cells is also semi-fluid and consists mainly of water, with the same substances dissolved in it. In addition, this cytoplasm has elements of a cytoskeleton, or a fibrous network that gives these larger cells shape and support. Now, the cell nucleus is the most obvious difference between the eukaryote and prokaryote. If you recall, in prokaryotic cells there was only a nuclear region and no membrane nucleus, no membrane-bound nucleus. But here in eukaryotes you have a nuclear envelope which is made up of a double membrane, each layer of which is structurally like the plasma membrane. Nuclear pores in the envelope allow RNA molecules to leave the semi-fluid portion of the nucleus, known as the nucleoplasm, and to participate in protein synthesis. Each nucleus has one or more nucleoli, which contain a significant amount of RNA. Also found in the nucleus of most eukaryotic organisms are paired chromosomes, each of which contain DNA and proteins called histones. The nuclei of eukaryotic cells divide by the process of mitosis, and prior to the actual division of the nucleus, the chromosomes replicate but remain attached, forming dyads. In most eukaryotic cells, the nuclear envelope breaks apart during mitosis, and a system of tiny fibers called spindle apparatus guides the movement of the chromosomes. During sexual reproduction, the nuclei of sex cells divide by a process of meiosis. After the chromosomes replicate, forming dyads, pairs of dyads come together, and during the course of the two cell divisions, the dyads are distributed to form 4 new cells, thus creating haploid cells. Now, the ribosomes of eukaryotic cells are much larger than prokaryotic cells and are made up of about 60% RNA and 40% protein. They have a higher sedimentation rate of 80S, and their subunits have sedimentation rates of 60S and 40S. Ribosomes are assembled in the nucleoli of the nucleus, and all ribosomes provide sites for protein synthesis, and some are arranged on chains as polyribosomes. Now, mitochondria are known as the powerhouses of eukaryotic cells, and these are exceedingly important organelles. They can be quite numerous in some cells and can even make up for up to 20% of the whole cell volume. Mitochondria are complex structures about 1 micrometer in diameter with an outer membrane, an inner membrane, and a fluid-filled matrix inside the inner membrane. Mitochondria carry out the oxidative reactions that capture energy in ATP. Energy in ATP is in a form usable by cells for their activities. Now, eukaryotic cells capable of carrying out photosynthesis will contain chloroplasts. They also have an outer and inner membrane. The inner stroma of chloroplast corresponds structurally with the matrix of mitochondria. However, unlike mitochondria, chloroplasts have separate internal membranes called thylakoids that contain the pigment chlorophyll, and this is the pigment that captures energy from light during photosynthesis. Both mitochondria and chloroplasts contain DNA, and they can replicate independently of the cell in which they function. This and other evidence has led many biologists to speculate that these organelles may have originated as free-living organisms. And here you can see a picture of the chloroplast. Again, the thylakoid membranes contain chlorophyll and other pigments and enzymes needed for photosynthesis. Thylakoids occur in stacks called grana, and grana are joined by membranous flat sheets called lamellae. Here you can see a colorized transmission electrons micrograph of a chloroplast from a leaf of a corn plant. Now let's discuss some of these membranous networks, including the endoplasmic reticulum and the Golgi apparatus. The endoplasmic reticulum, or ER, is an extensive system of membranes that form numerous tubes and plates inside the cytoplasm. The ER can be smooth or rough textured. The smooth endoplasmic reticulum contains enzymes that synthesize lipids, especially those used in making membranes, whereas the rough endoplasmic reticulum has ribosomes bound to its surface that give it a rough texture. The function of the rough ER along with the ribosomes to manufacture proteins. Vesicles from this system transport to the Golgi apparatus the lipids and proteins synthesized in or on the endoplasmic reticulum membrane. The Golgi apparatus is made up of a stack of membranous sacs, and the Golgi apparatus receives substances transported from the endoplasmic reticulum. It stores the substances and typically alters their chemical structure. It can package these substances into small segments of membrane called secretory vesicles. The secretory vesicles fuse with the plasma membrane and release secretions to the exterior of the cell. The Golgi apparatus also helps to form the plasma membrane and membranes of the lysosomes. Now, with regards to eukaryotic organelles and inclusions, you have lysosomes, vacuoles, peroxisomes, and cytoskeleton. Lysosomes are very small, membrane-covered organelles made by the Golgi apparatus in animal cells, and they can contain many kinds of digestive enzymes that could destroy a cell if those enzymes got released into the cytoplasm. Lysosomes fuse with vacuoles that form as the cell ingests substances, and they release enzymes that digest the substances in the vacuoles. Many bacteria that enter cells, especially those that were engulfed by white blood cells, are killed through this process. Now, peroxisomes are small membrane-enclosed organelles filled with enzymes as well. Peroxisomes are found in both plant and animal cells, but appear to have different functions in these two types of cells. In animal cells, their enzymes oxidize amino acids, whereas in plant cells they typically oxidize fats. Peroxisomes can convert hydrogen peroxide to water in both plant and animal cells. If hydrogen peroxide were to accumulate in cells, it would kill them, just as it kills bacteria when humans ingest it as an antiseptic, or use it as an antiseptic. Now, for vacuoles, in eukaryotic cells, vacuoles are membrane-enclosed structures that store materials like starch, glycogen, or fat to be used for energy. Some vacuoles form when cells engulf food particles. Water-filled vacuoles can add rigidity to plant cells. Now, regarding the cytoskeleton, this is a network of protein fibers made of microtubules and microfilaments. The cytoskeleton supports and gives rigidity and shape to a cell. It also is involved in cell movements, such as those that occur when cells engulf substances or when they make amoeboid movements. Now let's discuss motility in the eukaryotic cells. These cells can also have flagella, cilia, and pseudopods. Just like the external structures of prokaryotic cells, external structures of eukaryotic cells either assist with movement or provide a protective covering for the plasma membrane. Flagella in eukaryotes, which are larger and more complex than those in prokaryotes, are made up of 2 central microtubules and 9 pairs of peripheral microtubules in a 9+2 arrangement, surrounded by a membrane. Each fiber is a microtubule made of a protein known as tubulin. Eukaryotic flagella move like a whip, whereas prokaryotic flagella move like a rotating hook. One mechanism of eukaryotic flagellar movement is cross-bridging among dynein and other flagellar proteins. Through ATP hydrolysis, dynein plays a role in converting chemical energy in ATP to mechanical energy, which makes the flagella move. Flagella are most common among protozoa, but are also found in algae as well. Most flagellated eukaryotes have 1 flagellum, but some may have 2 or more. The only flagellated human cells are spermatozoa. With regards to cilia, these are shorter and more numerous than flagella, but they have the same chemical composition and basic arrangement of microtubules. These are found mainly in the ciliated protozoa, which may have up to 10,000 or more cilia distributed over their cell surface. Each cilium passes through a stroke and recovery cycle as it beats. And when the cilia of an organism beat in coordination, it can create a wave that passes from one end of the organism to the other, allowing them to move. The large number of cilia and their coordinated beating allow the ciliated organisms to move much more rapidly than those with flagella. Cilia of some cells can also propel fluids, dissolved particles, bacteria, mucus, and so on past the cell. Now, pseudopodia, or false feet, are temporary projections of cytoplasm associated with amoeboid movement. This kind of movement occurs only in cells without walls, such as amoebas and some white blood cells, and only when the cell is resting on a solid surface. When an amoeba first extends a portion of its body to form a pseudopodium, the cytoplasm is much less dense in the pseudopodium than other areas of the cell. As a result, cytoplasm from elsewhere in the organism flows into the pseudopodium through a process known as cytoplasmic streaming. An amoeboid movement is thus a slow and chain-along process. Now, regarding other aspects of cell wall components, a number of unicellular eukaryotic organisms have cell walls, none of which contain the peptidoglycan that is characteristic of bacteria. For example, algal cell walls are mainly made up of cellulose, but some others can contain polysaccharides. Cell walls of fungi consist of cellulose or chitin, or both. Protozoans have flexible external coverings called pellicles. Regardless of the composition, cell walls can give rigidity to protect them from bursting when water moves into them from the environment. Now, biologists believe that life arose on Earth about 4 billion years ago in the form of simple organisms, much like the prokaryotic organisms of today. However, fossil evidence seems to suggest that eukaryotic organisms arose only about 1 billion years ago. The way that prokaryote to eukaryote development took place is unknown, but endosymbiotic theory offers one possible explanation. As we've seen, the major difference between prokaryotes and eukaryotes is that eukaryotes contain specialized membrane-enclosed organelles, including a true nucleus. According to the endosymbiotic theory, the organelles of eukaryotic cells actually arose from prokaryotic cells that had developed a symbiotic relationship with the eukaryote to be. Symbiosis is a relationship between two different kinds of organisms that live in close contact. If one lives inside the other, the relationship is known as endosymbiosis. It's suggested that the first eukaryotic cell was an amoeba-like cell that somehow had developed a nucleus. Knowing the ease with which bits of cell membrane can pinch off to form vesicles, it's fairly easy to imagine that a primitive chromosome might have become surrounded by a membrane, thereby creating a rudimentary nucleus. The primitive eukaryote was probably a phagocytic cell, one that obtains its nutrients by engulfing material from the environment, including maybe other cells. The engulfed prokaryote may have been protected by a eukaryote, and the eukaryote could acquire some new abilities through the presence of such symbionts. Evidence supporting the theory of endosymbiosis comes from a comparison of the characteristics of eukaryotic organelles with those of prokaryotic organisms. For example, mitochondria and chloroplasts are approximately the same size as prokaryotic cells. Unlike other organelles, mitochondria and chloroplasts have their own DNA. Organelle DNA is present in the form of a single circular loop, like the chromosome of a prokaryote. Organelles have their own 70S ribosomes, which are like prokaryotic ribosomes, in contrast to the 80S ribosomes of eukaryotes. Organelle DNA and ribosomes carry out protein synthesis as it occurs in bacteria, rather than as it occurs when directed by nuclear DNA of modern eukaryotes. Antibiotics inhibiting protein synthesis by bacterial ribosomes do likewise on the ribosomes of the chloroplast and mitochondria. Mitochondria and chloroplasts divide independently of the eukaryotic cell cycle by means of binary fission. Also, the double membrane structures of mitochondria and chloroplasts strongly resemble the cell membranes of gram-negative bacteria, even having the same types of pores. And chloroplasts strongly resemble the structure of photosynthetic chlorophyll-containing prokaryotic cyanobacteria. And lastly, mitochondrial DNA most closely matches the DNA of the bacterium Rickettsia prowazkii. Here you can see an example of endosymbiosis. This is in the cytoplasm of Parasynympha, which is a protist that lives symbiotically in the hindgut of termites. Bacteria, shown here as dark ovals, act as mitochondria for the protist. And you can see here at the left of one that's actually dividing inside the protist. Now let's discuss ideas about movement across membranes. Living cells, whether they're prokaryotic or eukaryotic, are dynamic, and a cell is separate from its environment by a membrane across which substances constantly move in a carefully controlled manner. Understanding how these movements occur is essential to understanding how a cell functions. Very small polar substances, such as water, small ions, and small water-soluble molecules, probably pass through pores in the membrane. Nonpolar substances, like lipids and other uncharged particles, dissolve in and pass through membrane lipids. And still other substances are moved through the membrane by carrier molecules. Most large molecules are unable to enter cells without the aid of specific carriers. I want to go over a few simple concepts, the first of which is diffusion. Simple diffusion is the net movement of particles from a region of higher to lower concentration. For example, if you drop a lump of sugar into a cup of coffee, at first a concentration gradient exists with the sugar concentration greatest at the lump and least at the rim of the cup. Eventually, though, sugar molecules become evenly distributed throughout the coffee, even without stirring. Diffusion occurs because of random movement of particles. The length of time required for particles to diffuse across a cell increases with cell diameter. Materials can diffuse throughout small prokaryotic cells very quickly and throughout larger eukaryotic cells fast enough to supply nutrients and to remove waste fairly efficiently. If cells were much larger, however, diffusion through the cell would be too slow to sustain life. So diffusion rates may be responsible in part for the limitations of cell size. In this first figure, you can see an example of a simple diffusion illustration. Here, the random movements of molecules cause them to spread out or diffuse from an area of high concentration to areas of lower concentration until eventually they become equally distributed throughout the available space. In other words, they reach equilibrium. Now let's talk about osmosis. Osmosis is a special case of diffusion in which water molecules diffuse across a selectively permeable membrane. To demonstrate osmosis, you can start with two compartments separated by a membrane permeable only to water, as you can see here in this picture. One compartment contains pure water, and in the other compartment contains some large non-diffusible molecules such as proteins or sugars. Water molecules move in both directions, but their net movement is from pure water toward the water that contains other molecules. Thus, osmosis is the net flow of water molecules from a region of higher concentration of water molecules to a region of lower concentration across a semi-permeable membrane. Osmotic pressure is defined as the pressure required to prevent the net flow of water by osmosis. The least amount of hydrostatic pressure required to prevent the movement of water from a given solution into pure water is the osmotic pressure of the solution. Now let's discuss also active transport. In contrast to passive processes, active transport moves molecules and ions against concentration gradients from regions of lower concentration to ones of higher concentration. This is analogous to someone trying to roll a stone uphill, and it requires the cell to expend energy from ATP. Active transport is important in microorganisms for moving nutrients that are present in low concentrations in the environment of the cells, and it requires membrane proteins that act both as carriers and enzymes for the process. These proteins display specificity in that each carrier transports a single substance or a few closely related substances. Now let's talk about endo and exocytosis. In addition to the processes that move substances directly across membranes, eukaryotic cells can move substances by forming membrane-enclosed vesicles. These vesicles are made from portions of a plasma membrane. If they form by invagination or poking in of the membrane and surround the substance from around outside of the cell, the process is known as endocytosis or engulfment. If the vesicles inside the cell fuse with the plasma membrane and extrude their contents from the cell, then this is known as exocytosis. And you can see here an illustration of both processes of endo and exocytosis. Again, endocytosis being the process of taking materials into the cell, whereas exocytosis is the process of releasing them from the cell. Material taken in by the form of endocytosis is called phagocytosis and is enclosed in vacuoles known as phagosomes. The phagosomes fuse with lysosomes, which release powerful enzymes that degrade the contents of the vacuole. And these reusable components are then absorbed into the cell, and the debris is thrown out through the process of exocytosis. This is all I have for today's lecture. Thank you very much.